one. Which of the following are components of a nucleotide? A, base, deoxyribose or ribose, C, phosphate group, D, amino acid, E, fatty acid. Answer base, B, deoxyribose or ribose, C, phosphate group. Explanation: a nucleotide is composed of a nitrogenous base, a sugar, ribose or deoxyribose, and a phosphate group. Amino acids and fatty acids are not part of a nucleotide. 2. Which of the following statements about purines and pyrimidines is correct? A. Purines contain a two-ring structure. B. Pyrimidines contain a two-ring structure. C. Purines include adenine and guanine. D. Pyrimidines include cytosine, uracil, and thymine. E. Purines include uracil and thymine. Purines contain a two-ring structure. C. Purines include adenine and guanine. D. Pyrimidines include cytosine, uracil, and thymine. Explanation: purines are two-ring structures including adenine and guanine. Pyrimidines have a one-ring structure and include cytosine, uracil in RNA, and thymine in DNA. Uracil and thymine are not purines. Which of the following molecules is involved in purine nucleotide synthesis? A. Glycine, B. Glutamine, C. Thymine, D. Aspartate, E. Citrulline. Answer: A. Glycine, B. Glutamine, D. Aspartate. Explanation: amino acids such as glycine, glutamine, and aspartate are involved in the synthesis of purine nucleotides. Thymine is a pyrimidine, and citrulline is involved in the urea cycle, not purine synthesis. None of 4. Which of the following is the strongest type of base pair bond in DNA? A. AT, B. CG, D. AT, GT, AU. Answer B. Explanation: the CG base pair has 3 hydrogen bonds, making it stronger than the AT base pair, which has only 2 hydrogen bonds. This difference in bond strength contributes to the stability of DNA. 5. Which of the following is an example of a deamination reaction? A. Cytosine to uracil, B. Guanine to hypoxanthine, C. Adenine to hypoxanthine, D. Thymine to uracil, E. Cysteine to serine. Answer A. Cytosine to uracil, B. Guanine to hypoxanthine. Explanation: deamination refers to the removal of an amino group. Cytosine deaminates to uracil and guanine deaminates to hypoxanthine. Adenine deamination does not occur to hypoxanthine. Instead, it forms inosine in purine metabolism. 6. Which of the following molecules is necessary for pyrimidine synthesis? A. Glycine, B. Glutamine, aspartate, D. Thymine, E. methionine. B. Glutamine, C. Aspartate. Explanation: glutamine and aspartate are involved in the synthesis of pyrimidines. Thymine is a product of pyrimidine metabolism, not a precursor in the synthesis process. Methionine is involved in methylation reactions, not pyrimidine synthesis. Which of the following is the energy source for the formation of a phosphodiester bond in DNA replication? A. ATP, B. GTP, C. UTP, D. Triphosphate group of the incoming nucleotide, E. AMP. Answer: triphosphate group of the incoming nucleotide. During DNA replication, the energy for the formation of a phosphodiester bond comes from the triphosphate group of the incoming nucleotide, specifically the cleavage of a high-energy bond in the triphosphate. 8. Which of the following is the correct base pairing in DNA? A. AT, CU, GC, AT, CGTG, D. T, ACG. Explanation: In DNA, guanine pairs with cytosine GC and adenine pairs with thymine AT. The pairing of A with U occurs in RNA, not DNA. 9. Which of the following deamination reactions leads to a mutation? A. Cytosine to uracil, B. Adenine to hypoxanthine, C. Guanine to xanthine, D. Thymine to uracil, E. Cytosine to thymine. Cytosine to uracil. Explanation: when cytosine deaminates to uracil, uracil can pair with adenine instead of guanine, leading to a mutation in the DNA sequence. The other deamination reactions do not lead to the same mutation process. 9 or 10. Which of the following is a correct statement about the melting temperature of DNA? A. DNA with more AT bonds has a higher melting temperature. B. DNA with more CG bonds has a lower melting temperature. C. DNA with more CG bonds is more stable. D. AT content does not affect DNA stability. E. GC pairs have fewer hydrogen bonds than AT pairs. Answer C. DNA with more CG bonds is more stable. Explanation: CG pairs have 3 hydrogen bonds, making them more stable than AT pairs, which have only 2 hydrogen bonds.